Hello, fellow seekers of neural knowledge. Today, we're venturing deep into the intricate crossroads of the brain, where signals dance and information flows seamlessly. Welcome to the fascinating world of central nervous system synapses. We are Equation by pharmacist moms, and together, we're about to unravel the mesmerizing complexity that makes communication within the brain nothing short of a symphony. In this video, we'll explore the microscopic yet mighty structures that enable neurons to communicate, the central nervous system synapses. From neurotransmitters to synaptic clefts, get ready for a journey that unveils the secrets of how our thoughts, movements, and emotions are orchestrated in this neural dance. Whether you're a neuroscience enthusiast, a student eager to grasp the fundamentals, or simply curious about the wonders of the brain, this video is tailor-made for you. So, hit that subscribe button, join our community of brain explorers, and let's dive headfirst into the captivating world of central nervous system synapses. Before we get started, don't forget to give us a thumbs up if you're ready to embark on this synaptic adventure, and now, let's unlock the mysteries that lie within the heart of our neural connections. In this video, we will learn 1. Transmission of information in central nervous system. 2. Types of synapses, chemical and electrical. 3. One-way conduction at chemical synapses. 4. Physiologic anatomy of the synapse. 5. Presynaptic terminals. 6. Mechanism by which an action potential causes transmitter release from the presynaptic terminals, role of calcium ions. 7. Action of the transmitter substance on the postsynaptic neuron, function of receptor proteins. 8. Ion channels. 9. Second messenger system in the postsynaptic neuron. 10. Excitatory or inhibitory receptors in the postsynaptic membrane. Let's begin. How is information transmitted in the central nervous system? Let's study. Information is transmitted in the central nervous system, mainly in the form of nerve action potentials, called simply nerve impulses, through a succession of neurons, one after another. However, in addition, each impulse may be blocked in its transmission from one neuron to the next, may be changed from a single impulse into repetitive impulses, or may be integrated with impulses from other neurons to cause highly intricate patterns of impulses in successive neurons. All these functions can be classified as synaptic functions of neurons. How many types of synapses are there? There are two major types of synapses: the chemical synapse and the electrical synapse. First one are chemical synapses. Almost all the synapses used for signal transmission in the central nervous system of the human being are chemical synapses. In these, the first neuron secretes, at its nerve-ending synapse, a chemical substance called a neurotransmitter, or often called simply transmitter substance, and this transmitter in turn acts on receptor proteins in the membrane of the next neuron to excite the neuron, inhibit it, or modify its sensitivity in some other way. More than 40 important transmitter substances have been discovered thus far. Some of the best known are acetylcholine, norepinephrine, epinephrine, histamine, gamma-aminobutyric acid, that is, GABA, glycine, serotonin, and glutamate. Electrical synapses, in contrast, are characterized by direct open fluid channels that conduct electricity from one cell to the next. Most of these consist of small protein tubular structures, called gap junctions, that allow free movement of ions from the interior of one cell to the interior of the next. Only a few examples of gap junctions have been found in the central nervous system. However, it is by way of gap junctions, and other similar junctions, that action potentials are transmitted from one smooth muscle fiber to the next in visceral smooth muscle, and from one cardiac muscle cell to the next in cardiac muscle. Next is one-way conduction at chemical synapses. Chemical synapses have one exceedingly important characteristic that makes them highly desirable for transmitting most nervous system signals. They always transmit the signals in one direction, that is, from the neuron that secretes the transmitter substance, called the presynaptic neuron, to the neuron on which the transmitter acts, called the postsynaptic neuron. This is the principle of one-way conduction at chemical synapses, and it is quite different from conduction through electrical synapses, which often transmit signals in either direction. Think for a moment about the extreme importance of the one-way conduction mechanism. It allows signals to be directed toward specific goals. Indeed, it is this specific transmission of signals to discrete and highly focused areas, both within the nervous system and at the terminals of the peripheral nerves, that allows the nervous system to perform its myriad functions of sensation, motor control, memory, and many others. Upcoming is physiologic anatomy of the synapse. This figure shows a typical anterior motor neuron in the anterior horn of the spinal cord. It is composed of three major parts: the soma, which is the main body of the neuron, a single axon, which extends from the soma into a peripheral nerve that leaves the spinal cord, and the dendrites, which are great numbers of branching projections of the soma that extend as much as 1 millimeter into the surrounding areas of the cord. As many as 10,000 to 200,000 minute synaptic knobs, called presynaptic terminals, lie on the surfaces of the dendrites and soma of the motor neuron, about 80 to 95% of them on the dendrites, and only 5 to 20% on the soma. These presynaptic terminals are the ends of nerve fibrils that originate from many other neurons. Many of these presynaptic terminals are excitatory, that is, they secrete a transmitter substance that excites the postsynaptic neuron. But other presynaptic terminals are inhibitory, they secrete a transmitter substance that inhibits the postsynaptic neuron. Neurons in other parts of the cord and brain differ from the anterior motor neuron in the size of the cell body, the length, size, and number of dendrites, ranging in length from almost 0 to many centimeters, the length and size of the axon, and the number of presynaptic terminals, which may range from only a few to as many as 200,000. These differences make neurons in different parts of the nervous system react differently to incoming synaptic signals and, therefore, perform many different functions. Now, we will learn the presynaptic terminals in detail. Electron microscopic studies of the presynaptic terminals show that they have varied anatomical forms, but most resemble small round or oval knobs and, therefore, are sometimes called terminal knobs, boutons, end feet, or synaptic knobs. This figure illustrates the basic structure of a synapse, showing a single presynaptic terminal on the membrane surface of a postsynaptic neuron. The presynaptic terminal is separated from the postsynaptic neuronal soma by a synaptic cleft, having a width, usually of 200 to 300 angstroms. The terminal has two internal structures, important to the excitatory or inhibitory function of the synapse: the transmitter vesicles and the mitochondria. The transmitter vesicles contain the transmitter substance that, when released into the synaptic cleft, either excites or inhibits the postsynaptic neuron, excites if the neuronal membrane contains excitatory receptors, inhibits if the membrane contains inhibitory receptors. The mitochondria provide adenosine triphosphate, that is ATP, which in turn supplies the energy for synthesizing new transmitter substance. When an action potential spreads over a presynaptic terminal, depolarization of its membrane causes a small number of vesicles to empty into the cleft. The released transmitter, in turn, causes an immediate change in permeability characteristics of the postsynaptic neuronal membrane, and this leads to excitation or inhibition of the postsynaptic neuron, depending on the neuronal receptor characteristics. What is the role of calcium channels in the release of the transmitter from the presynaptic terminals? Let's discuss. The membrane of the presynaptic terminal is called the presynaptic membrane. It contains large numbers of voltage-gated calcium channels. When an action potential depolarizes the presynaptic membrane, these calcium channels open and allow large numbers of calcium ions to flow into the terminal. The quantity of transmitter substance that is then released from the terminal into the synaptic cleft is directly related to the number of calcium ions that enter. The precise mechanism by which the calcium ions cause this release is not known, but it is believed to be when the calcium ions enter the presynaptic terminal, it is believed that they bind with special protein molecules on the inside surface of the presynaptic membrane, called release sites. This binding, in turn, causes the release sites to open through the membrane, allowing a few transmitter vesicles to release their transmitter into the cleft after each single action potential. For those vesicles that store the neurotransmitter acetylcholine, between 2,000 and 10,000 molecules of acetylcholine are present in each vesicle, and there are enough vesicles in the presynaptic terminal to transmit from a few hundred to more than 10,000 action potentials. What is the action of the transmitter substances on the postsynaptic neuron? The membrane of the postsynaptic neuron contains large numbers of receptor proteins. The molecules of these receptors have two important components. A binding component, which protrudes outward from the membrane into the synaptic cleft, here it binds the neurotransmitter coming from the presynaptic terminal. And an ionophore component, which passes all the way through the postsynaptic membrane to the interior of the postsynaptic neuron. The ionophore in turn is one of two types. An ion channel, which allows passage of specified types of ions through the membrane, or a second messenger activator, which is not an ion channel but instead is a molecule that protrudes into the cell cytoplasm and activates one or more substances inside the postsynaptic neuron. These substances in turn serve as second messengers to increase or decrease specific cellular functions. We will learn the ion channels and the second messenger activator in succession, in detail. The ion channels in the postsynaptic neuronal membrane are usually of two types. Cation channels. They most often allow sodium ions to pass when opened, but sometimes allow potassium and/or calcium ions as well. The cation channels that conduct sodium ions are lined with negative charges. These charges attract the positively charged sodium ions into the channel when the channel diameter increases to a size larger than that of the hydrated sodium ion. But those same negative charges repel chloride ions and other anions and prevent their passage. When cation channels open and allow positively charged sodium ions to enter, the positive electrical charges of the sodium ions will in turn excite this neuron. Therefore, a transmitter substance that opens cation channels is called an excitatory transmitter. Anion channels. They allow mainly chloride ions to pass, but also minute quantities of other anions. For the anion channels, when the channel diameters become large enough, chloride ions pass into the channels and on through to the opposite side, whereas sodium, potassium, and calcium cations are blocked, mainly because their hydrated ions are too large to pass. Opening anion channels allows negative electrical charges to enter, which inhibits the neuron. Therefore, transmitter substances that open these channels are called inhibitory transmitters. When a transmitter substance activates an ion channel, the channel usually opens within a fraction of a millisecond. When the transmitter substance is no longer present, the channel closes equally rapidly. The opening and closing of ion channels provide a means for very rapid control of postsynaptic neurons. Next are second messenger system in the postsynaptic neuron. Many functions of the nervous system, for instance, the process of memory, require prolonged changes in neurons for seconds to months after the initial transmitter substance is gone. The ion channels are not suitable for causing prolonged postsynaptic neuronal changes because these channels close within milliseconds after the transmitter substance is no longer present. However, in many instances, prolonged postsynaptic neuronal excitation or inhibition is achieved by activating a second messenger chemical system inside the postsynaptic neuronal cell itself, and then it is the second messenger that causes the prolonged effect. There are several types of second messenger systems. One of the most common types uses a group of proteins called G proteins. This figure shows in the upper left corner a membrane receptor protein. A G protein is attached to the portion of the receptor that protrudes into the interior of the cell. The G protein in turn consists of three components: an alpha component, that is the activator portion of the G protein, and beta and gamma components that are attached to the alpha component, and also to the inside of the cell membrane, adjacent to the receptor protein. On activation by a nerve impulse, the alpha portion of the G protein separates from the beta and gamma portions, and then is free to move within the cytoplasm of the cell. In this figure, for subsequent possible effects of the G protein are shown, including opening an ion channel in the membrane of the second neuron, activating an enzyme system in the neuron's membrane, activating an intracellular enzyme system and/or causing gene transcription in the second neuron. Let's learn them in detail. Opening specific ion channels through the postsynaptic cell membrane. Potassium channel that is opened in response to the G protein. This channel often stays open for a prolonged time, in contrast to rapid closure of directly activated ion channels that do not use the second messenger system. Activation of cyclic adenosine monophosphate, that is CAMP, or cyclic guanosine monophosphate, that is CGMP, in the neuronal cell. Either cyclic AMP or cyclic GMP can activate highly specific metabolic machinery in the neuron. Therefore, it can initiate any one of many chemical results, including long-term changes in cell structure itself, which in turn alters long-term excitability of the neuron. Activation of one or more intracellular enzymes. The G protein can directly activate one or more intracellular enzymes. In turn, the enzymes can cause any one of many specific chemical functions in the cell. Activation of gene transcription. This is one of the most important effects of activation of the second messenger systems, because gene transcription can cause formation of new proteins within the neuron, thereby changing its metabolic machinery or its structure. Indeed, it is well known that structural changes of appropriately activated neurons do occur, especially in long-term memory processes. It is clear that activation of second messenger systems within the neuron, whether they be of the G protein type or of other types, is extremely important for changing the long-term response characteristics of different neuronal pathways. What are the excitatory or inhibitory receptors in the postsynaptic membrane? Some postsynaptic receptors, when activated, cause excitation of the postsynaptic neuron, and others cause inhibition. The importance of having inhibitory, as well as excitatory, types of receptors is that this gives an additional dimension to nervous function, allowing restraint of nervous action and excitation. What are the different molecular and membrane mechanisms used by the different receptors to cause excitation or inhibition? Let's learn. Different molecular and membrane mechanisms used by the different receptors to cause excitation include opening of sodium channels to allow large numbers of positive electrical charges to flow to the interior of the postsynaptic cell. This raises the intracellular membrane potential in the positive direction, up toward the threshold level for excitation. It is by far the most widely used means for causing excitation. Depressed conduction through chloride or potassium channels, or both. This decreases the diffusion of negatively charged chloride ions to the inside of the postsynaptic neuron, or decreases the diffusion of positively charged potassium ions to the outside. In either instance, the effect is to make the internal membrane potential more positive than normal, which is excitatory. Various changes in the internal metabolism of the postsynaptic neuron to excite cell activity or, in some instances, to increase the number of excitatory membrane receptors, or decrease the number of inhibitory membrane receptors. Different molecular and membrane mechanisms used by the different receptors to cause inhibition include: opening of chloride ion channels through the postsynaptic neuronal membrane allows rapid diffusion of negatively charged chloride ions from outside the postsynaptic neuron to the inside, thereby carrying negative charges inward and increasing the negativity inside, which is inhibitory. Increase in conductance of potassium ions out of the neuron allows positive ions to diffuse to the exterior, which causes increased negativity inside the neuron. This is inhibitory. Activation of receptor enzymes that inhibit cellular metabolic functions that increase the number of inhibitory synaptic receptors or decrease the number of excitatory receptors. And there you have it, our deep dive into the captivating world of central nervous system synapses. We hope this journey has shed light on the incredible complexity and precision that underlies every thought, every sensation, and every movement we experience. If you found this exploration of synaptic connections intriguing, be sure to give it a thumbs up, share it with fellow neuroscience enthusiasts, and subscribe for more enlightening content on the marvels of the brain. There's so much more to uncover. Thank you for joining us on this synaptic adventure. Your curiosity fuels our exploration of the vast and mysterious terrain of the central nervous system. Keep those questions coming, stay engaged, and, most importantly, keep marveling at the wonders within your own mind. We are Equation by Pharmacist Moms, signing off for now, but stay tuned for our upcoming videos as we continue to unravel the complexities of the brain. Until next time, keep your neurons firing and your curiosity thriving.